Hello and welcome everybody to our webinar today, which is called Far Beyond Any Control or Prediction, the Convergence of NGTs and AI, Risks to Biodiversity. Thank you all for joining. I think this webinar comes at a very crucial moment because there is a vote foreseen in the European Parliament tomorrow on the trilogue outcome on new genomic techniques applied to plants. So I believe it's very good moment also to make political decision makers aware of the dynamics that are created by this convergence of genetic engineering with artificial intelligence and the risks that come along with this. We are recording our webinar and we will probably publish it later. The decision still needs to be taken. And just to give you a quick overview over the program. So we first will have a presentation of our new report, and then Dr. Then will explain the implications for the regulatory process. After that, Professor Tielbörger will focus on risks, especially for wild plants. And after that, Dr. Reeves will give an outlook into future deregulation initiatives on genetically engineered microorganisms. And then we close with with Jim Thomas, who will explain about the economics of gene editing, who is standing to profit from these technologies. And of course, there will be time foreseen for discussion after each presentation and also at the end. At the bottom of the screen, you will find the question and answer button where you can hand in your questions. So without any further ado, I would like to give the word to my colleague, Dr. Matthias Juhas, who is a research officer at Testbiotech and who will present our new report. Please go ahead.
Yes, thank you, Astrid. I will now try to share my slides. And I hope you can see them.
Yes, perfect.
Perfect.
Thanks.
Okay, so thank you, Astrid, for the nice introduction. In my presentation, I will give a rough overview about the current developments in the convergence of artificial intelligence and new genomic techniques and the resulting risks for biosafety, that is, risks for the environment and biodiversity. Let's start with the status quo or the implications of the convergence of AI and genetic engineering, which is currently attracting a great deal of attention, as it not only promises great benefits like wealth and prosperity, but also holds considerable risk potential in terms of disruption and instability. In the risk debate, there are many concerns, especially about biosecurity, which is the misuse of AI and NGT, but also economic aspects are becoming more and more important since the US and China in particular are engaged in political and economic competition over AI and NGT. And with the planned deregulation of NGT plants, the EU is also taking a stance in the competition for global leadership in this field. But unfortunately, what is largely absent from all the discussions are the risks to the environment and biodiversity, meaning biosafety. That's why they are in the focus of the new report. The convergence of AI and NGT was made possible primarily by two technological developments. First, in recent decades, large amounts of biological data have been digitalized and made available in databases, and AI can browse vast amounts of this big data in seconds for the design of new gene variants and gene combinations. On the other hand, a wide range of new genomic tools and methods made it possible to genetically engineer almost any gene in any life form. And this rapid pace of technological development is also leading to ambitious announcement, where high expectations are being raised with the promise that AI, NGT, and big data analysis enables greater precision, efficiency, and sustainability, or breeding 5.0 will enhance food productivity, improve food quality, and fosters resilience to climate change, including bold claims like smart crops and intelligent-driven speed breeding. However, although AI is now routinely used in the planning of genetic engineering experiments in plants, there are only still a few examples of NGT plants in which AI has played a decisive role in their development. And one example for this are low lignin NGT poplars, where AI was used to design and evaluate around about 70,000 different genome editing strategies to reduce lignin content in NGT trees. In this example, up to 6 genes were altered simultaneously with the so-called multiplex CRISPR editing, with the intended genetic changes falling below the threshold of 20 for category 1 NGT plants, and the resulting low lignin poplars are supposed to potentially be more suitable for wood processing. For these low lignin NGT1 poplars, which would be exempt from risk assessment under the EU's deregulation proposal, an environmental risk assessment should definitely be mandatory, as it is unclear what effects these genetic alterations will have on the tree's interactions with other species or their response to environmental stress. Poplars naturally grow in sensitive ecosystems, including the black poplar, and if NGT poplars enter these natural populations through pollen, seeds, cuttings, or broken branches, their retrievability would be a serious problem, or perhaps impossible, as poplars produce millions of pollen grains and seeds. Furthermore, natural poplar populations could be endangered or displaced, including the species that rely on them. In the next example, NGTs were used to exchange or to change the architecture of flowers in tomatoes and soybeans to facilitate robotic pollination. And here too, less than 20 genetic changes were sufficient to manipulate the flowers in a way that they produce exerted female stigmata and become male sterile to develop a robot-friendly flower morphology. Robotized cross-pollination should then be carried out by AI-trained robots. And although these roboplants are primarily designed for AI-designed assisted breeding in greenhouses, they have nevertheless already been tested in the fields. From plants, let's now move on to livestock, where there are only a few publications on AI and NGT so far, because the focus is currently on programs for conventional livestock breeding. But as there are meanwhile also a lot of NGT applications in livestock, it is to be expected that AI and NGT will become increasingly important in this area too. And one trend here is the alteration of gene regulation, since, for example, the Roslin Institute in Edinburgh, which you maybe know from Dolly the sheep, is currently involved in cattle breeding projects in which AI is used to identify regulatory gene variants. And in order to achieve this goal, they have developed AI and NGT strategies that can be used both in cattle and other species. They write, with the increased availability of high-quality training data, it is expected to further improve the performance of livestock models in prioritizing novel functional variants and ultimately improving advanced breeding approaches. However, the most prominent at present are the so-called de-extinction projects by a US company, Colossal Biosciences, which, with the help of AI and NGT, aim to bring extinct animals such as woolly mammoths, direwolves, dodos, Tasmanian tigers, and moas back to life. And to achieve this, in case of the woolly mammoth, they want to change over 60, they call it cold weather genes, that are, for example, responsible for shaggy hair, and mice were already manipulated to show longer and curly hair like mammoths. In case of the direwolf, which was recently featured in the Time magazine, by altering 20 targets in the genome of a gray wolf, they created an NGT wolf that is stronger and larger as their natural counterparts and has a white coat. However, what the company is actually creating or wants to create, besides media hype, are genetically engineered versions of existing species, such as Asian elephants, gray wolf, South American running birds, and marsupial mice. These animals would also pose environmental risks, shown here exemplarily for the NGT direwolf. If they were to escape into natural gray wolf populations, they could also reproduce. And since they are larger and stronger than their natural counterparts, they and their offspring would have a fitness advantage and could displace natural populations. This would have significant consequences, not only for the protected wolf species, but also for ecosystems and possibly humans if the genetic changes also lead to altered hunting behavior. Unexpected consequences could also arise in the midterm if, for example, the NGT animals were more susceptible to diseases. These traits could also spread along with the wolves, posing an additional risk to the species survival. So these projects could therefore achieve the exact opposite of what they claim to be aiming for. Instead of leading to the revival of extinct species, they can endanger the survival of species that exist today. Concerning AI and NGT insects, various databases now offer the possibility of searching the genetic material, including that of model organisms, plant pests, or pathogen vectors. And in this context, AI and NGT is, for example, used to find target genes that are suitable for insecticidal applications using RNA interference, to identify regulatory sequences that can be used to alter gene regulation, to predict the outcome of CRISPR-Cas interventions, and to optimize the process of genetic engineering. With the so-called gene drives, which are used to create GMOs for example, combat invasive species, artificial gene constructs can spread much faster in subsequent generations than through normal inheritance. And here, AI and NGT are used to identify suitable target genes to be engineered via the artificial gene drive constructs and to assess the impact of the gene drives on the target populations. Adding a few words on AI and NGT in microorganisms. Already for several years, NGT bacteria have been commercially marketed mainly in the US as fertilizers to improve, for example, nutrient uptake in crops. And now the company Bayer also aims to establish itself in the so-called biologicals industry with AI and NGT through a collaboration with Ginkgo Bioworks. And according to statements in their databases, they have access to more than 300,000 agronomically relevant bacterial strains and more than 2.7 billion metagenome datasets. The release of AI and NGT microorganisms such as these biologicals is also problematic since there is often a lack of reliable criteria for robust risk assessment. Possible risk scenarios here include that NGT traits may become unstable and be transferred to other species, and that reproduction and spread of microorganisms is almost impossible to control. Coming now to the last example, last year the first AI and NGT designed functional viruses were created that have never been existing in nature. These AI viruses have superior properties compared to the template strain of so-called bacteriophages. For this, the AI genomic foundation models EVO1 and 2 were trained with almost 3 million genomes, also taking complex interactions between genes and their regulation into account. So this example shows that AI and NGT could be used to synthesize entire biological systems and create completely novel properties. But some of the new variants cannot only reproduce successfully with the help of bacteria, they can also overcome resistance and spread with greater effectiveness. So in terms of the risks, bacteriophages can continuously evolve, acquire new properties, and, for example, kill bacteria that are essential for human life, plants, and animals. Furthermore, this approach could also be applied to viruses that spread dangerous diseases. Although in this example, the AI was not trained with data from human pathogens, the approach of these experiments could technically be transferable to viruses that are dangerous to humans. And that's why many experts are warning about the dangers of malicious applications or even military use. So to sum this all up, AI and NGT allows engineering of organisms to be made in extremely short periods of time. This represents a much greater intervention in the characteristics of species and their ecosystems than could ever be expected from breeding or natural processes. In the course of evolution, only a certain selection of characteristics is realized. Now, the convergence of AI and NGT can bring about everything simultaneously and in all species. And that's why the release of NGT organisms has the potential to disrupt entire ecosystems and become a new cause of species extinction. And last not least, the convergence of AI and NGT can lead to major leaps in development, but the possibilities for controlling this development are lagging dramatically behind. And with this, I'd like to thank you for your attention and look forward to the discussion.
Yes, thank you, Matthias. I would now already like to jump to the next presentation, and then maybe Matthias get back to you later again. Christoph Then, who is the director of Testbiotech, will explain what are the implications of this convergence of AI and gene editing on the regulation, so on the political regulations.
Yeah, thanks for your interest. And I will ask my colleague Mario Kudruf to start my slides. And indeed, I will try to focus now on NGT plants and the use of AI and the consequences for regulation, just because of the ongoing discussion and upcoming decision-making upon future regulation of NGT plants. So I start with some ideas about what gene scissors can do, and it's very clear that gene scissors can, just as CRISPR-Cas, can overcome many constraints which are there in conventional breeding and restrict the results in conventional breeding. These gene scissors are recombinant enzymatic mutagenes that can introduce gene variants that were previously unknown and also unlikely to result from conventional breeding methods. And we have many examples where we see just very small numbers of changes that is enough, that in combination to achieve genotypes and also phenotypes that cannot be achieved by conventional breeding. And these genotypes and resulting NGT plants, from the perspective of health and environment, have to be considered as new to the environment, and consequently they have to be subjected to risk assessment. And indeed, there are many environmental risks already described in scientific literature in regard to NGT plants, such as negative effects on pollinators and food webs, increased invasive potential, weakening of natural plant population, depression in yields, toxicity for insects, unintended effects on soil organisms, and endangerment of protected species. And just recently, we published a report on manipulated flowers and NGT plants, and there we also discuss environmental risks such as for food security, for natural populations, and for pollinators. So when it comes now to the plant regulation, there seems to be an idea, as originally produced by the European Commission, that there is something like a safety threshold for a number of mutations. So in future, there would be a threshold of 20 genetic changes which would become a central part of future regulation. And according to current estimates, more than 90% of all NGT plants then would fall within this category of plants which do not deserve mandatory risk assessment. And in result, NGT plants, even if they pose environmental risks, such as, for example, the poplar tree, which we just saw, could be marketed and released without environmental risk assessment. Next slide. So when it comes to the examples we just heard, we see that AI can do a lot in plants. And we also heard already that all these plants would be below the threshold of these 20 genetic changes. So they might release into the environment without risk assessment, with one exception, which is the genetically engineered insecticidal maize, which was also published in peer review as designed by artificial intelligence. This would not go without risk assessment because just it's producing insecticidal proteins. But the other NGT plants do not pose less risk than this specific maize. So this differentiation between these plants with 20 genetic changes or below 20 genetic changes does not seem to be based on sound science. So with artificial intelligence, now please go back, in place, for the background of these 20 genetic changes, it's very clear that artificial intelligence could be used to design specific NGT plants which do not exceed the proposed thresholds, but nevertheless pose severe environmental risks, as these examples show. And these NGT plants could nevertheless then be marketed and released without environmental risk assessment. So let's go to the next slide. When we take a broader view, we could say NGTs can cause a crack in creation, as a title of a book from Jennifer Doudna in 2017 was introducing the CRISPR-Cas machineries. In any case, NGTs are a disruptive technology, as we were informed by the US company Corteva, which is the world's most important player in patent applications on NGT plants. But the specific problem with this disruptive effect is, of course, that these are concerning our livelihoods and the future biodiversity in Europe. So it took a million years to adapt a butterfly to a specific flower, but disrupting the ecosystems by using NGTs and AI might just take a few decades. We should be aware that this is a real and major risk for the future of our biodiversity in Europe. Next slide. So the proposed regulation with the 20 genetic changes just tries to create the impression that certain rules and safety standards will continue to apply to the release of NGT plants. In reality, however, industry is just giving a carte blanche for importing and releasing the plants via fast-track approval. Therefore, we come to the conclusion that with the planned NGT regulation, the European Union would be about to hand over its responsibilities for health and environment to the free market forces, which of course would be contrary to basic ideas and legal foundations of the European Union. Next slide. So what to do, how we can protect biodiversity in Europe? Well, as a first step, we think it's easy to say that at least those NGT plants which inherit genotypes that were previously unknown and are unlikely to be obtained from previous breeding methods, as we just saw some examples, if this must become a trigger for mandatory environmental risk assessment. And this general principle is especially relevant for plants that can persist in the environment and reproduce with wild plant species. So for the sake of future generations and homeland biodiversity, we think the proposal for deregulation of NGT plants has to be rejected. Next slide. So maybe you're not aware, we also were not aware before we published our report on manipulated flowers, to which extent already now wild species are involved in NGT applications. All the species you see in here were already NGT applications, and all of these plant species also can persist in the environment and perform gene flow to other populations of the same species or of species which are related to them. Next slide. So finally, I also want to draw your attention to a long-term perspective. Of course, NGT plants is just a small part of the discussion. We are proposing 10 measures how to contain the risk of AI and biotech, which are basically coming from the basic ideas are coming from other experts in the field. But we think we should adopt this also to risk to biodiversity and biosafety. So like promoting risk research or expertise in civil society or the precautionary principle and an expanded technology assessment are definitely things we have to work on and agree on also internationally. Next slide and final slide. So for further reading, I recommend these reports, which you will find on our website. And of course, these reports will then offer further references that you can follow. Thank you very much.
Yes, thank you, Christoph. But now I would suggest we go back to the ecological risks of deregulation. And we have Professor Dr. Katja Tielbörger with us today. She is a professor for ecology at the University of Tübingen in Germany, and she will explain about risks to ecosystems and especially also to wild plants. So please, Katja, we're very glad to have you here with us today again, and you can go ahead.
Thank you, Astrid. Yeah, I'll be narrowing down the discussion a bit. So previously it was about AI and NGTs and many types of organisms, and I'll be more focused on the specific proposal that has been agreed upon in the trilogue and the ecological risk. And I'm not only speaking here on behalf of the University of Tübingen, which is my home university, but I'm also the speaker of the working group New Genomic Techniques of the German, Austrian, and Swiss Ecological Society, the largest ecological society in Europe. And within that society, within that working group, we have issued an opinion paper related to the proposal of the EU Commission back then in 2023. And here you can see the link. And one of the important citations from that report, from that discussion paper, was that we write that new genomic techniques are an attempt to solve problems on a high organizational level, and that is on an ecological level with methods on a molecular level. Why do we think it's a problem? Because if we look at the scales at which ecologists are working, and I'm talking about scientists when I say ecologists, we work on a cellular and molecular level too, just as molecular biologists do, but we're mostly working from an individual level, across community level, up to the entire biosphere. And the topics we are dealing with, they almost read like a wish list for what is actually written in the Commission's proposal. So NGTs are promised to address many of these environmental problems, such as nutrient cycling, climate change, ecosystem functions. We're talking about novel organisms, a topic very relevant for the release of NGTs into the environment, protecting biodiversity, increasing yield in agroecosystems, and many more. So these are the topics that ecological science deals with. Now, when we look at where the promises are made in the proposal and where the expertise is that actually does these promises, we see that there's a huge mismatch. So the scientists, when you read in this proposal and also in the media, it always says science says that NGTs will be saving the world, basically. So solving all the problems that you can see here on the right-hand side. But when you look at who these scientists are, they're mostly scientists working on a molecular level. So there's a big gap between the people talking about the promises and the expertise they actually have. So there's a lack of ecological expertise, scientific ecological expertise when it comes to assessing both the risks and the benefits. And this is why we issued this opinion paper related to the proposal that is currently still maybe at least for one day under debate. And this mismatch between the scientific expertise actually going into the proposal and the expertise that I think is needed for assessing the benefits and the risks of this technique has led to a whole range of blind spots in the current proposal. And for example, a major blind spot is about the risks and the benefits. In the current proposal, the risks and the benefits are only defined by the genetic changes that are imposed on a particular plant. And this idea of that a certain number of changes in the genome, which is 20 by 20, to categorize these types of plant into plants that are assumed without having any ecological risk or environmental risk is complete scientific nonsense. So this threshold doesn't make sense from an ecological point of view, because both the risks and the benefits are actually determined by the phenotype of the plants, because it's the phenotype of the plant that interacts with the environment. And the same applies not only for the risks, but also for the benefits. So there's lots of promises about the benefits, but there's literally no scientific evidence that actually supports this idea of these tremendous benefits of new genomic techniques for plant breeding. Now, where do these risks come from? So the first step, and we heard this also in the previous talk, is that a novel organism gets into the environment. How does it get into the environment? It might be outcrossing with neighbors, with related plants from the same species or closely related species. And there's lots of review studies by now, one of the conclusions of these studies is that the movement of transgenes beyond their intended destinations, for example, an agricultural field, is a virtual certainty. And here you can see just a few pictures of plants where it has been shown repeatedly that these plant species can outcross with either their own species or relative species that are related to them in the wild. And the two species in the red boxes are two species that have been used for old genetic engineering methods, and they have been found in the environment. So outcrossing of plants that are new, whether they are genetically engineered or just new in a different way, is basically a certainty. So what happens when these plants outcross in the wild? And there's actually two possibilities. The first thing is when they outcross into the wild, this is also called genetic pollution. And there's also been a very recent scientist warning on genetic pollution, meaning that there are novel genotypes in the environments. And this can have two different outcomes. And one of them is greatly overlooked. This outcome is called outbreeding depression. And it's the fact that genes enter the environment that are actually detrimental for natural populations. Because when people think about environmental risk, they usually think this novel organism is going to spread like hell and causing problems because it spreads aggressively. But I think the inverse of that is introducing genes that are detrimental for the fitness of wild plants, is actually much larger. And we've heard this example of the poplar. I mean, introducing a species which is also a wild species into agriculture that has a low lignin content will be much more wobbly, less well defended against pathogens. There's also applications in poplars where they changed the flowering time, which means we would have a complete mismatch in flowering time among the introduced genotypes and the natural genotypes. So these effects, they're called outbreeding depression, and they actually lead to a decreased fitness of wild organisms. And because this is the case, there are actually national and international laws in place that protect the environment from these effects of outbreeding depression. So introducing novel genotypes in the environment is actually not allowed when we look at national and international laws. So the current proposal is at odds with these legislations. So genetic pollution, on the one hand, can have detrimental effects due to swamping with maladapted genotypes, but obviously there's also the effect that these new novel genotypes can be aggressively spreading. And we have lots of evidence from invasion ecology that shows that the more often you try, so the more larger is the number of novel organisms that enter a new environment, the larger is the likelihood that they become invasive and actually spread aggressively. This is almost a no-brainer. I mean, it's basically statistics. The likelihood of a single organism to cause problems is rather low, but the more organisms, novel organisms you introduce, the larger is the risk. So the sheer number of novel organisms that will certainly enter the environment with the new genomic techniques, and even more so if we combine new genomic techniques with artificial intelligence, the sheer number, this quantitative difference, translates into a qualitative one. So the risk of these novel genotypes spreading aggressively is increasing with the new genomic techniques. We also know from literature that the novelty of the phenotype determines whether or not a genotype becomes aggressive. And the new genomic techniques have the power to basically manipulate everything and create plants that are very different from what they were before. And we also know from invasion biology that any introduction of a novel genotype into the environment is entirely unpredictable. We don't know what will happen, and it's irreversible. We cannot bring these genes back. And this is why, as ecologists, we always call for the precautionary principle and a case-by-case risk assessment. Now, another blind spot is related to this this blunt statement of saying new genomic techniques are basically exactly the same as classical breeding, which doesn't make sense. I've talked about the large numbers, which makes them very different from classical breeding and which translates into a qualitative difference when it comes to risk assessment. And they are also completely novel genotypes. So we can create with NGTs genotypes that have not been there before and cannot be created by breeding. But the major difference between NGT and classical breeding, at least in the proposal, is that the proposal allows a deregulation of NGT1 for all plant species. And this is a major blind spot. So each time I'm talking to my friends or even scientists and colleagues about this, everybody stares at me and says, oh, I've never heard this before. But in fact, it says in the proposal, this proposal applies for all plant species, with a very weird exception, which is red algae. In wild species, when you bring wild species into the wild, the likelihood of outcrossing is not only a likelihood, but is a certainty. And that creates an unprecedented danger to wild populations, communities, and ecosystems. I mean, we're talking about estimates of 300,000, maybe 500,000 wild plant species. And so then often people ask me, so what's so bad about wild species? I mean, where are these applications? And there's many applications already discussed in the ecological community. For example, on the level of the IUCN, there's been a very lively and year-long discussion about should we use synthetic biology methods in nature conservation, yes or no? And this lively debate, which has a very equivocal outcome, has not even been considered in the design of the new regulation on an EU level. So the ecologists, the scientists have been completely ignored. We have thought about this issue already, and there might be positive applications in nature conservation, but they should only be allowed when there's a previous risk assessment. For example, if we want to combat invasive species. The de-extinction also, I mean, Christoph has touched upon this, is complete nonsense. I mean, species go extinct for a reason, and as long as we don't reduce as long as we don't remove that reason for extinction, we're not going to de-extinct the species. And if I look at the sheer amount of money that goes into this, sorry to say, nonsense research, if we would take that money and put it into nature conservation, we would be saving so many more species. And then there's also applications, and nobody thinks about those, that I call just for fun applications. So there's do-it-yourself kits for CRISPR-Cas that you can download from the internet. And then there's the application in research. Many researchers, ecologists, molecular biologists are not working on agricultural plants. We're working on wild plants. And this powerful technology has created, a momentum currently in the research, and everybody wants to work with it and put their stuff out in the field. And nobody looks at that. And the weird thing about this application to all species is that it's not only highly, highly dangerous, it's not even needed. So if we look at why this new deregulation has been proposed, it's all about applications of NGT for food and feed. Nobody talks about applications in nature. So the extension of this proposal to wild plants is not only highly dangerous, but it's not even relevant, raising the question why it has been introduced. There's no history of safe use for genetic modification or breeding of 300 or more wild plant species and their introduction into the wild. So this idea of equivalence to breeding is completely immaterial for wild populations. So one of the major conclusions of our opinion paper of the GFO is that we call for a clear protection of wild plant species from any type of deregulation. And that is a case-by-case risk assessment prior to release into the wild, consistent with a precautionary principle, which is basic to European legislation. And finally, a last blind spot related to the proposal is this promise that we're going to save the world. So if we look at this scheme of the planetary boundaries, all these ecological, environmental catastrophes that our planet is facing, there are all these promises that with NGTs we're going to solve this problem. We're going to create climate adaptive plants that are drought resistant without losing any yield. This is scientific nonsense. Plants that have a higher drought resistance will automatically have lower yield. There's this promise that these techniques will be much faster. And I agree with the fact that the effects of the techniques will be much faster, especially with the use of AI. But I mean, even those plants that are bred for agriculture, they will have to pass through a whole number of legislative steps that won't make them that much faster. There's this promise that the plants will be saving the world because we will be introducing pathogen resistance, but also pathogen resistant trades off with yield. And also, if we grow plants in a monoculture that are resistant to pathogens, it means that resistance of the pathogen to the resistance of the pathogen of the plant will be evolving much faster. So this is no solution. So also these promises of the NGTs for, I call it saving the world, there's no solid scientific evidence, and in many cases, even scientific evidence against these promises. So the blind spot is basically towards the true solution that we already have at hand. So there's basically thousands of studies in ecology and agroecology that already show us how we can make our agricultural system more sustainable. And one of the main measures is to increase biodiversity in agriculture, to have more varieties in a field or even more than a single species on a field. We know that increasing biodiversity in our landscape will increase the resistance to drought, the resilience to drought, the stability of the yield. It will increase productivity. It will increase pathogen resistance, and you just name it. There's so many things that are promoted by just increasing biodiversity. And with this debate that we've been leading for so many years now about the new genomic techniques, this has completely distracted from the true solutions that can be implemented instantly and without any risk. So to summarize, I call it science-based recommendation, and science here means ecology, the risks and benefits of the current proposal. So this current proposal is blind to fundamental scientific evidence about both benefits and risks on the level at which the NGTs will be applied. So you have to consult ecologists for risk assessment and benefit assessment, and it hasn't been done. The hand-waving quantitative molecular criteria for distinguishing between NGT1 and NGT2 are completely immaterial for deregulation, because environmental risk is a function of the phenotype and must be evaluated case by case. Our most important point in our opinion paper is that deregulating NGT1 for all plant species is a serious threat for biodiversity conservation and sustainability. So if this proposal will be voted in favor for tomorrow, hopefully not, we'll actually be voting for something that has the potential to have exactly the opposite effect of what we were dreaming of. So the true solution is diversification. Diversification guarantees yield stability, low environmental impact, high resistance and resilience, and social justice. So my appeal would be to the EU that the EU should be spearheading a true agricultural revolution with science-based, fast and efficient solutions to the global poly crisis, instead of focusing on technological solutions that are based on promises and no scientific evidence. Thank you very much.
Yes, thank you, Katja, for your passionate talk. I would now like to move to the presentation of Dr. Guy Reeves, who used to be with the Max Planck Institute for Evolutionary Biology, but who is now with Save Our Seeds. And you will give an outlook on what is coming on genetically engineered microorganisms. As far as I understand, we have now a proposal on the table by the Commission dating back from December last year. But you will give us more details. Please go ahead.
Thanks, Astrid. Actually, I'm not going to give a lot of details about that proposal, but I am going to mention its implications. So first, I'm going to start off with the plant NGT proposal. Don't worry, you don't need to read any of the text. But I just this is just to illustrate that the original proposal And then what is being decided on now is rather different. And the yellow means things that were present in both, in the original and the current ones, and dark blue means things that have been introduced during the negotiation process. And this is just to show how much the technical language ended up changing. There are actually more changes, and this, I'm going to argue, is partly to do with AI and the realization that using vague and non-scientific language in the era of AI, with its power to come up with multiple solutions to complex problems, means that we need to be aware of how important language is. That doesn't mean technical language, particularly. That doesn't mean that we can skip the part where, I mean, a poor policy, however well drafted, is still going to be a poor policy. So the NGT plant proposal changed. It mostly became more restricted during the process, which I think, and one of the reasons it became, particularly with respect to not producing insecticidal plants as part of the NGT1 class, the least regulated class, was probably negotiations, but it also was some publications and discussions directly with the Commission or DG Sante. And these are a few of them. And one of them was also done by Testbiotech. So having a longer consultation period allows for the improvement of laws, even if we're not that happy with the outcome. So this new proposal which came out in the 16th of December last year, just before Christmas, it shows that plants are just the beginning of this sort of new technology deregulation, and this is for this proposal is for deregulating microbes or a class of microbes and the processing of organs for transplants. This is very unusual to bundle two completely separate topics in one proposal. And the only explanation is given is that there was an urgent political need to, that's why they couldn't produce the normal documents, explanatory documents and risk impact assessments, which is very strange, and it's rather bad democratic practice to include completely different proposals together, and presumably maybe even hope that they will stay together and it will be difficult to vote against one. For some reason I'm not advancing. Okay, all right. So I'm going to focus on just a small part of the technical proposal of this, the microbe part, and I'm going to ignore the transplants for human medical treatment, treating patients. So this is about the creation of a new class called low-risk genetically modified microbes, and it has 3 criteria, but all the text in blue is very vague. What's of concern, what's well characterized, why use organisms instead of the normal species. This is sort of an example of the kind of proposal you really shouldn't make in the age of AI. You should be much more specific, because if the text was to stay this way and the proposal was accepted, AI could likely make it apply to much more than the lawmakers ever imagined. So I'm going to give one example of microbes, and Matthias mentioned some, and this is a slightly different one. These are actually at least one of them is for sale now. These is engineering plasmids, the natural conjugative plasmids that bacteria share amongst each other between species. And these can often carry CRISPR proteins.
Sorry, Guy, to
Yes.
interrupt you,
Yeah.
but I just have somebody telling me from the audience that they cannot see your presentation. Now I don't know.
I can
Could
see it.
I can see it as well, but one of the participants told me. Okay, well, it's other participants can see it, so I think it's fine.
Okay.
I'm sorry to interrupt you.
No problem.
Please go on.
So yeah, this is a product you can see that's sold currently in Brazil, which uses the plasmids have been genetically modified, so the CRISPR proteins no longer target their normal bacteriophage viruses, but they're targeting another bacterial species, in this case a Salmonella species. So you're engineering bacteria release into the guts or the environment to target other bacteria by genetically manipulating plasmids that are shared amongst them. So this is a fairly uncontrolled way to do this. Interestingly, in Brazil, this is approved as being not a GMO technique. But so there's another company which proposes to spray these bacteria across fields. Other companies propose to sell them as probiotics for animals to eat or even humans to eat. So would such approach, given the technique, given the proposal that came out in December, I just thought it would be a bit of fun to see whether this fairly radical approach could actually fit this new light touch regulation or no regulation approach. And they would have to switch bacteria. At the moment, the Brazilian one is using E. coli, but if it used Bacillus subtilis, which is a very common thing for probiotics, yes, it would fit A. Yes, it would fit B because it's on this list. And C, would it fit those criteria? Well, it's difficult to say. It all depends, interprets your word concern, but maybe. So these are quite radical proposals to basically, on the tail end of the plants, to do something very similar for microbes. But microbes are even more hard to recall than plants, and they're even really quite difficult to detect once you release them into the environment. So that's basically my point. Is in the age of AI, we shouldn't be giving up on coming up with good policies. We definitely shouldn't skip that, but it's important that the language that is regulations is that people are aware of AI's power to go around these and effectively deregulate and massively expand these categories beyond what was imagined. And yeah, that's what that, and with that we do need more, possibly more than ever, solid deliberative processes which can't be accelerated too much. Yeah, that if they are accelerated, and this proposal seeks to accelerate it on the back of a needed transplant modifications to some regulations, this would be not a very good process for democracy, particularly as some of these microbial techniques, including the probiotics, are likely to be perceived by people as a no opt-out technology. It'll be very difficult to avoid once there is large-scale adoption or spreading of these things in the environment. And that's where I'll stop.
Okay, great. Thanks, Guy, for making that point. We already have the first question for you. Do you have any idea why they are bundling together GMO microbes with human transplant regulation in the EU?
I am fairly unfamiliar with this, but people tell me that this is really, really unusual to do this, and that the aim might be to keep them together to prevent proper scrutiny of the NGT proposal and its implications. Well, the fairly uncontroversial change to how long organs can be used before they're transplanted into humans. Maybe it's a way to make it more difficult for members of parliament to vote against or even delay or amend this proposal. But if that's what it is, that seems like a fairly cynical and not good for democracy.
Yes, we also have one national expert commenting on that. He's saying that the lumping together of different policy topics does also have the consequence that the overall proposal is likely not discussed in the Council working group with an environmental focus, but in the Council Working Party on Pharmaceuticals and Medical Devices, where discussion of the GMM proposal seems to be ill-placed, to put it mildly. So
that seems like an even more informed point. I mean, one thing that's interesting is at least for this low risk category, there's this existing list called the Qualified Presumption of Safety list, which I think 133 species are on that list, and those. Though their applications that they're used for are very broad. So it's industrial processes, food processes, agriculture, and also probiotics for humans and agriculture. So yeah, it would be very strange to do this. This bundling is not what a healthy democratic institute would want to do, and I'm sure Michael Eckersdorfer is right that this would be a way to limit the scrutiny, which I think I was trying to argue is essential to get buy-in or to produce safe regulations.
Yes, well, maybe there's still a way to split it up if Parliament and Council want to do so. But for the moment, I'd like to thank you for your presentation. I don't have other questions directed to you directly right now. And I would like to go now to Jim Thomas, who is a European AI and Society Fund Fellow running his own consultancy, which is called scanthehorizon.org. And he formerly used to be with the etc. group. And he's going to give a talk about the economics of gene editing and explaining who is driving this technological convergence and who stands to profit from it. So we're very much looking forward to your talk, Jim. Please go ahead.
Hello, thanks very much, Astrid. Can everybody see the presentation okay?
I think so. I can see it in any case.
Great, super. And it's been really, really wonderful listening to all these presentations and information, and the report is extremely helpful. I was asked to make some comments on the report, and I would definitely start there. You know, that first of all, much of the conversation around so-called AI X bio or generative biology, this coming together of genetic engineering and AI, looks and the hype is all around medical applications, production of drugs, production of materials. So it's helpful to have a report that looks very focused on the agricultural applications. And I think that's a useful corrective and a good lens through which to see what's happening in this field of convergence. And I also particularly appreciate that this report has really focused on the biodiversity risks, and we've heard some of that very clearly just now from Katja, but also from Guy, and in fact before that from Matthias and from Christoph. given that at the international level, the place where regulation and oversight sits is the Convention on Biological Diversity, there has been a long process on oversight of synthetic biology, which is where this convergence sits and where NGTs sit. And indeed, there was a very clear proposal, which was supported by European Union, to have an in-depth assessment on the impacts of the convergence of AI and biotech and synthetic biology on biodiversity, on the objectives of the Convention on Biodiversity, and that was cut out. That was cut out very much by Argentina, Brazil, New Zealand, Canada, and as a result, we haven't had the conversations that need to happen about how this convergence impacts biodiversity. So this starts to fill in some of what would have been happening at an international level, and there's still a chance it could happen if the European Union at the next SBSTA meeting of the CBD, which is in Nairobi in June, and also in the fall at the COP, once again push for this assessment that we really urgently need. Mostly what I'm going to talk about, though, just is on the question of the sort of economic promises. Let me see if I can. Yeah, so my background, or what I've been doing for the last year, is I've been part of a fellowship for the European Artificial Intelligence and Society Fund, trying to understand the economic and market power implications of having big AI companies moving into the genetic engineering space, synthetic biology, gene editing, and so forth. And so I'm just going to talk a bit to that, because I'd say the biggest, the TLDR, if you like, the key thing that I've learned looking at this field in the last year is that really the value in this convergence is not about the products, really, and it would be a mistake to look at the products. The value really lies in the data processing that is done by large companies such as Google, NVIDIA, Oracle, and the public relations value right now to be able to talk about speculative but one day real products that may exist or may not, such as drugs and crops and things. That's really where the value is. We heard at the beginning of this webinar from Matthias, where he quoted, actually, I don't know if it was intentional quote, this promise by Mustafa Suleyman that this convergence has great benefits around increased prosperity and wealth. And if I have one criticism of the report that Testbiotech brought out, it's that maybe it relies a little bit too much on some of the things that Mustafa Suleyman wrote in his book, The Coming Wave. So Mustafa Suleyman is the CEO of Microsoft's AI. He was one of the founders of Google DeepMind. He's a sort of industry leader. He really is not a scientist or even like a very good social scientist or anything like that. He's somebody whose whole career has been convincing wealthy people to give him money. And I think there's a sense in which what's going on in the hype around AI and biotech it is about selling and supporting the current generative AI industry. And I'll talk more about that in a moment, that these assumptions that are being promulgated to policymakers that this is somehow going to bring a lot of wealth to Europe or to countries are very misplaced. This is really about keeping alive a bubble that urgently needs investment. Because the thing that is certainly clear is that when you look across the players in this area of AI biotech, the companies who are making the models, they are the large AI companies, whether it's Microsoft with Mustafa Suleyman, he came from Google, who were doing the same thing, Meta, NVIDIA, Oracle, Alibaba, Salesforce, OpenAI. All of these extremely large tech titans appear to be moving in to this area. Notably, none of these are European. At the same time, the European institutions are concerned about digital sovereignty. There is a worry that as we move into bio-digital era, we're once again ceding control to basically large American corporations and a couple of Chinese corporations. This is not an area that's giving any power, underlying power, to European industry. That's not happening here. In my study, I was looking at what is the scheme that the industry runs on, and this is broadly it. And I don't need to go over all of this right here, but I think it's helpful just to see a few things about how the AI biotech industry is coming together. At the heart of it, of course, you have these platforms, some of which you heard about from Matthias and others, that are redesigning genetic material. These the largest of these, like for example, Evo2, which is supported by NVIDIA, has 10 trillion tokens. It sounds like a very large thing, but these require a lot of infrastructure. This is why we're seeing, in the AI space, this build out of data centers across Europe, across North America, now moving into Africa and Latin America. The level of computing power and therefore machines that are required is immense, and who's benefiting from this is actually the largest investor in this space, which is NVIDIA. And so, at the heart of this, there's a very expensive technical build out. AI is already costing us over $7 trillion. It's the largest industrial build out since the building of the railways in the middle of the 19th century. So even without the biotech side of it, this is a very expensive enterprise, and it's highly concentrated. But in order for all this to work, you require data. And so what's also happening is an attempt to try and grab data, that those 10 trillion pieces of data that are used in some of these models are just a drop in the ocean of the genomic information that's out there. And more importantly for AI, if you don't have a representative sample, then it's useless. Your whole project is really going to be based on bad science, and we'll have hallucinations and errors, which is what we see in AI all the time, but we're seeing it now very much with AI biotech. And so there's the need to build out a massive project in sequencing, in collection. We have companies like Basecamp Research in the UK who are trying to go to countries to grab as much new DNA as they can, a new biopiracy rush. This is also required for any of this to work, if you believe that you can scale up to make a working model. But unlike the sort of genetic engineering, so unlike the AI that we see with ChatGPT, you don't just print up a picture on a page and you see it immediately, or a text on a page. You actually need to have labs to keep iterating. And so the physical side of this, the need to scale up labs, the need to scale up the sort of test design cycle over and over again is also immense. And that's a massive infrastructure cost. So on top of the $7 trillion that's being built up for AI, and what's going to end up being, I think, trillions of dollars for trying to grab the data, you're going to have further trillions of dollars required in order to make the labs to even just test this to see if it works. And that's why there's a big grab to try and get European Union funds, to try and get military funds, to try and get philanthropic funds. This is not ultimately, when you begin to take apart this is not a business model that's cheap. It's incredibly expensive. And it basically says, if you can just throw more computing power at this, eventually we'll get there. But they need to keep getting that money needs to keep coming in. And so, this is about the AI industry saying very clearly, we've got drugs coming, we've got crops coming, this is going to happen. And by giving the impression that something is happening there, by putting some little bit of money into this AI biotech, they can keep the largest spigot of money going towards artificial intelligence. And we've been here before. Those of us who remember the announcements at the turn of the millennium around the Human Genome Project will remember that Bill Clinton and Tony Blair stood up with the leaders of the Genome Project and said, this is it. We've used computing to crack the genome, and now we're going to be able to cure diseases like Alzheimer's, Parkinson's, diabetes, cancer. Bill Clinton says it's conceivable our children's children will know the term cancer only as a constellation of stars. He expected that would happen in the next 10 years. We're now 25 years down the line, and In fact, you know, Alzheimer's has doubled since 2000, cancer rates have doubled. That's also true about diabetes. None of this was true. The underlying technology didn't actually work, but there was a tremendous amount of hope and hype, and we're seeing this again with AI. In my interviews with senior synthetic biology leaders trying to understand what it means to have these AI companies come in, it's become clear to me that those within the industry or within the science know perfectly well that this is speculative. And I really appreciated this comment by someone who I can't say who it is, but they're very senior in the synthetic biology world. Don't underestimate people's excitement and enthusiasm for doing the easy thing, especially if it costs lots of money. Because if it costs lots of money and it's easy, suddenly you're gifted an opportunity to organize lots of money and people, and you get this coalescing into centralized concentrations of power. There's some people who actually believe AI will solve all problems, including that it will help us understand biology well enough to engineer it, but I don't believe that. There's things AI can't get to because we don't know how to get to them anyway, if it's right. If we don't actually make biology easier to engineer, well, guess what? It won't be easier to engineer. But what I mostly notice is the politics of power. And AI is now a sandbox for the politics of power. It's captured the attention of people who love power. And I think this would be my caution, that the hype that there is large amounts of wealth somewhere around the corner through the use of AI to make new NGTs and release them, not only is it not actually relevant to Europe, it's large American mostly and Chinese companies doing it. Not only does the basic technology not work because it doesn't work, but that this is all being used as a smokescreen for investment. And it would be problematic if on the basis of trying to support that investment, we then threaten our biodiversity, our health, and our future. So I would stop there. Thank you.
Yes, thank you, Jim, for this interesting talk. Well, I think the takeaway from the webinar is better stick to the precautionary principle. Vote against deregulation if you can. Does somebody want to have a final word before we close the conference? Maybe Christoph, Katja, Matthias. Katja, are you still there? You're always very passionate. Do you want to have a last word?
Oh my. Yeah, stop it. I mean, what can I say? I'm not against new genomic techniques, but this proposal for deregulation is just bound to lead to the opposite of what it was actually wanted to achieve. So yes, that's my wish. I hope that my wish will come true.
Yes. Yes. Maybe we can close it here. We also hope this wish comes true. And some people said we should clone you, Katja. We should use NGTs to clone you. Would that maybe be a solution?
I wouldn't want to meet myself in person, but yeah, I don't know. Thank you.
All right. Well, I wish you all a very nice day. Keep on smiling and keep on fighting.